Today I'm going to be walking you through the Genetically Modified Information Lab from Kansas Corn STEM. Hi, my name is Lacey and I'm a lead teacher for Kansas Corn STEM Education. Today we're going to be walking through the genetically modified information activity that you can do with your students. The purpose of this activity is to get your students to kind of get a working definition of what they think is a GMO and then apply that to our scientific concepts that we go through in our classroom on a regular basis. So what you're going to need to get started is really simple supplies that you already have in your classroom. So some note cards, something to write with, maybe a highlighter, and you're ready to go. The very first thing that I like to do is I like to get our students thinking about what do they know about GMOs. Activate that prior knowledge. And there's a really cool video on our website that you can take a look at as well. Here's a hint. This is what it might look like. Basically, after you watch that video, students will kind of have in their mind what they might already know about GMO. And what we're going to do is get them to write it down. So the first thing that they're going to do is they are going to write their own definition of what they think GMO means on a note card. Pretty simple. Give them time to do this, give them plenty of time to think, and then time to write. That's a critical part of this. The next thing they're going to do is find a partner. They're going to compare their definitions. And any words that they have that are the same, they're going to go ahead and highlight. Those are common words that we find across all definitions, but specifically with this partner or this team. The next thing is you have them go through those same definitions and figure out if there are any words that can go away. Are there any words here that we don't need, that are extra words or that don't really get our point across? What are the words that are critical and must stay? After you have students go through that, you'll have some words that are crossed out, some that are highlighted, and some that are circled that mean they have to be there. When you're finished, have them come up with an operational definition that they can all agree on. Generally, this can take 10 to 15 minutes, but it really gets students thinking about what are things that this definition has to have, and it brings all of their points of view together so that they can come up with a working definition for everyone. Now here's the big question. Why is a working definition so important? Take a look at this. Operational definitions are used all the time, all over the place. Here's a great example, the analogy of the blind men and the elephant. So the blind men all approach the elephant from different positions. They can feel the elephant, but they can't see him. So the one that might walk up to the tusk might say it's a spear. The one that gets to the ears might say it's a fan, or a tail, the rope. But everyone brings their own point of view because they have their own perspective. We have to take into consideration all of the background that our students already have walking into our classroom. And it's even more powerful if they can come up with an operational definition that they work out on their own. You can guide them through this, and they can come up with that definition. For the purposes of today, we're going to work with this definition. Our operational definition is that GMO means to intentionally change a living thing's original genes or DNA. That's what we're going to use today, and that's what you can use in your class. As we walk through, we're going to use that information to really help our students kind of parse through some different techniques that we use to breed animals and specifically plants. Now, as you go through, we can start modifying things. What does modifying mean? Well, this is where genetics comes in. When you talk about your genetics units and you talk about Punnett squares or crosses or hybrids, that's all modification. It's just modifying from one generation to the next. And so being able to walk your kids through this also helps you bring in all of those genetic terms that you've been using for the last unit. The very first thing that you're going to need for this part of the activity is you're going to need these cards. So these cards all are about different types of breeding. We have interspecies crosses, selective breeding, mutagenesis, transgenesis, simple selection, and genome editing. 6 cards, but with powerful actions. First step, have your students segregate these cards into 2 groups. The first group, non-GMO. The second group, GMO. Give them time to read the cards, talk amongst themselves, and see what they come up with. Generally, having them work as a pair or a group increases the amount of talk you're going to have too. So as we go through, in a lot of classrooms, what we end up with is we get a lot of students thinking that simple selection and things like interspecies crosses and hybridization are non-GMO. They put all the rest over here. So let's take a closer look. When we think about the history of genetic modification in crops, we go way far back. 10,000 years ago is when we first started domesticating crops. And when we did that, we were simply selecting what traits we wanted to see in our organisms. We wanted to see grains that had more production, more yield. We wanted to see crops that were more resistant to drought. And so we took those with us as our civilizations moved from place to place. So simple selection is a way that we can change things up. Another example is that interspecies crosses. We would take those with us and we would find other places with native plants that would allow things to work together. So technically, as we work our way through, things kind of need to change. So let's look back at our cards. So these cards with simple selection, we're still choosing what traits we want to see. But if we weren't there, maybe Mother Nature wouldn't have come up with that combination. So we changed those genes. Same thing with interspecies crosses. By taking a plant from one location and putting it somewhere else, we are transplanting those genes. If we hadn't been there, those changes wouldn't have happened. So technically, as we look at our cards, we take all of our cards from non-GMO to GMO, specifically looking at that definition of modification. Now, if your students are thinking, wait, that's not possible, direct them to this. Before domestication, a lot of the crops that we look at as common foodstuffs today look nothing like their ancestors. As we go through and look at things, you can have your students branch out and look up things like bananas or eggplants, even carrots and cabbage. As they go through, as we have changed things, we have developed new traits and exaggerated those traits and what we come up with. My very favorite one to look at is, of course, the corn. Teosintite to corn is a huge journey. So does that change your sorting? Now we've got non-GMO over here, kind of empty, and all of our GMO over here. But wait, they're not all the same. So what do we do now? Now we need to talk about these traits in a different way. We're going to use 2 new words. Instead of non-GMO and GMO, we're going to look at traditional and genetic engineering. Now, repeat the process. Sort which ones you feel like fall under the traditional breeding categories and genetic engineering. Give your students plenty of time to work this out. They might have to come up with an operational definition for what genetic engineering means or what traditional means, but make sure that you guide their discussions. As we go through, you might also want to give them some more information to help them better understand this. Here we go. Methods of plant breeding. If we want to go into this definition, traditional usually means that we're taking 2 genomes, combining them together, and hoping for a good outcome. We have no control over what genes get changed, and we have no control over the outcome. We are letting the genetic process of reproduction basically throw the dice, and we hope for a great outcome. From those good outcomes, we can then exaggerate those beneficial traits. Genetic engineering, on the other hand, is where we're targeting a specific gene. It could be from a different donor organism or from a same type of organism. We just want that gene to ensure that it is in the end product of our crosses. So now let's think about these as one gene versus many. Once you walk through that definition with students, now we can kind of point out a little hint on our cards. So if I look down here at the bottom of my cards, it says genes, how many are affected? So if we're talking about one gene or a few, then we're going to go under genetic engineering. If we're talking about more than one gene, we're going to go under traditional. That's going to be our sexual reproduction. Our immense variation that is created by that process is going to be seen through traditional breeding processes. So from there, you might see something that looks more like this. Specifically, we're going to look at these as our traditional processes. We have no idea how many genes are going to be changed, and we're really just hoping for a good outcome. Over here with genetic engineering, we're looking at one or a few genes in some cases, and even just one. So as we go through, walk your students through this process. A lot of kids look at this and they confuse the word genetically engineered with genetically modified. And that's really where we have to break that misconception. Here's another example. Once you have it sorted, have them take a look at this. So where would you draw the line here? Selective breeding, we can see, has 10,000 to 300,000 possible genes being affected. But remember, we want to go all the way down to one or a few. So as we walk our way from left to right, we can see interspecies crosses, that has quite a few. Mutagenesis, that's random, unknown, probably thousands. Transgenesis, there we go, 1 to 3. So that's going to be our genetically modified. Now, only transgenesis, as we look through this, from that example, was an example of a genetically engineered organism. We'll talk more about genome editing a little bit later. One of the things that students always like to see is what are some of the combinations that we can create through transgenesis, and why did we do it? There's a great activity for us to do for that too. It's speed dating. What kid doesn't like the idea of becoming a little bit more familiar with a GMO? In this process, we're going to use a different set of cards. These cards all have organisms on one side and information on the back. And what you will see is that some cards have donor, while others have recipient. These cards are available on our website, so you can print off your own copy if you don't have them already. What you're going to do is shuffle up these cards and distribute them to your students. Try and make sure that you are separating those pairs as you go all the way through the classroom. You can always eliminate some of the options if you need to, or if you want to make it a little bit more challenging, look at some of the ones that have more than one partner. As you walk through, students will have to work to pair a donor and a recipient together. As they do that, they'll be reading through some of the information that they see on the cards. Information here is vitally important to students understanding which pair to go together. Give them time to read their cards individually, separate into donors and recipients in the classroom, and then let them go. Have them find their pair, come up with their reason, and figure out if they're correct. From here, there's lots of things you can do. You can have students figure out what organisms are involved, who the donor is, what are they giving, who's the recipient, why do they need these genes, are these the same types of organisms, what problem is being addressed, and how can this actually help the world or make things a better place. From there, students can then present their information in lots of different ways. One of my favorite is to do a gallery walk. Have students create their own poster where they can tape up the cards but list out that information. Post those up around the room and let students walk all around figuring out what information they feel is most important. From here, students could also do lots of other things. If you really wanted to challenge them, or if you need some distance learning, you could also limit the number of cards that you give them access to. Print off only a few pages, or print them as a PDF. You could send those to students, and then they would have to figure out which donor and which recipient go together from that limited amount. Another example of something you could do from this is you could have students go through the entire life cycle of the creation of this transgenic organism. GMOanswers.com has some great examples for this. Here you can see we walk through the process of trait identification, transformation, greenhouse testing, regulatory science, which is a lot more than kids think it is, field testing, and getting the seeds to farmers. When it comes down to it, kids don't really know as much as they think they do about genetically modified organisms, and it's our job to help teach them. One of the last and most valuable things that I like to show my students is this diagram right here. Again, this is from GMOanswers.com, but it really shows the genetic traits that are expressed in GMOs that are found in the United States. You can use this as a jumping off point to talk about the labeling versus non-labeling controversy as well. When you see a can of green beans that says that it's non-GMO, did we really need that label on there in the first place? What about that Himalayan sea salt? Does it need a label? So as you go through, talk to your students about what organisms actually have genetically modified traits and which ones don't. Last but not least is that very, very important genome editing. Genome editing, this guy right here, is the forefront of what we are doing today to really understand about how genetic modification is being done. CRISPR-Cas9 is a great way to help students really further this debate. Have them look up some articles, figure out whether it is GMO or non-GMO, and make up their own mind.